Hi friends, today we are going to see the past year NEET questions for the chapter Molecular Basis of Inheritance, and today in this video we are going to complete part 2. So initially we have completed part 1, so those people who haven't gone through this part 1, kindly go through so that this will help you in the continuity with the chapter. So when it comes on NEET aspects, you can expect at least 2 to 3 questions from this chapter, so kindly concentrate on this chapter called Molecular Basis of Inheritance. So let's begin with the first question, which was asked in the year 2007. The length of DNA molecule greatly exceeds the dimensions of the nucleus in eukaryotic cell. How is this DNA accommodated? So we're talking about the nucleosomes here. The nucleosome model explains the packaging of the histone proteins and DNA in the chromatin material which forms the chromosome. Therefore, what it is accommodated in, how is the DNA accommodated? It is a supercoiling that is found within the nucleosomes. Therefore, the right answer to this question is option A. Next question, a sequential expression of a set of human genes is called as dash. Very easy question for you to answer. Yes, it is called as DNA sequence or DNA. A sequential expression of a set of human genes is called as DNA sequence. So what does a sequence have? The basis? Yes, it has a basis there. Next question. During transcription, RNA polymerase holoenzyme binds to the gene promoter and assumes a saddle-like structure. What is its DNA binding sequence here? And this question was asked in the year 2007. So before we go into the explanation part, let's go to and let's discuss few points about transcription. What do you mean by transcription? See, transcription and translation, you must have heard about it. Transcription is the first of the several steps of the DNA-based gene expression in which a particular segment of DNA is copied into the RNA by the enzyme called as RNA polymerase. Both DNA and RNA are nucleic acids which use the base pairs of the nucleotides as a complementary language. So what happens here in transcription? It is a process in which the gene's DNA sequence is copied, transcribed to make an RNA molecule with the help of the enzyme called RNA polymerase, and this uses one of the DNA strands, that is the template strand, as a template to make a new complementary RNA molecule. Transcription ends in the process called termination, which is the last step of the process called transcription. So now, what do you think will be the answer here? I've given you a clear explanation of what is transcription. The right answer is the DNA binding sequence of RNA polymerase is called as TATA, T-A-T-A box, TATA box, which is C. Next question, the two polynucleotide chains in DNA are dash. Very easy question. How are they? They are usually antiparallel. Okay, they are antiparallel. The two chains are antiparallel, that is, if one end goes from 5' to 3', the other end would start from 3' to 5'. Therefore, the right answer is option B. Next question, which antibiotic inhibits the interaction between DNA and mRNA during bacterial protein synthesis? So this you might not be aware of. The antibiotic used here is neomycin. Okay, neomycin is the antibiotic which will inhibit the translation of the bacterial so that it can affect the host cell. Therefore, the right answer is does not affect the host cell. I'm sorry in this part. So which antibiotic is used? Neomycin. It's an antibiotic which inhibits the translation of the bacterial cell so that it cannot affect the host cell. Therefore, the right answer is option A. Next question here, DNA template sequence. So you have a sequence here. It is transcribed over mRNA as dash. You just need to match and analyze the statements here. Any idea what it is? So the right one is option B, because we have C, T, G, A, T, A, G, C. This goes with guanine. We have adenine, cytosine, then we have uracil, then we'll have yeah, I think I'm right, we'll have adenine, uracil, it will be cytosine and guanine. Therefore the right answer is option B. During transcription, from the DNA template, complementary RNA is formed and thymine is replaced with uracil because we are associating with RNA. Next question. Extranuclear inheritance occurs in dash. So what do you mean by extranuclear inheritance? So extranuclear inheritance is also called as cytoplasmic inheritance, and this is a transmission of genes that occur outside the nucleus. It is found in most of the eukaryotes and commonly known to occur in the cytoplasmic organelles such as mitochondria and chloroplast, or sometimes in the cellular parasites like viruses and bacteria. So extranuclear inheritance occurs in the chloroplast and mitochondria, which is option B. Sometimes it is also found, this mechanism is also found in parasites like viruses and bacteria as well. Therefore the right answer for this question is option B. Next question. Which form of RNA has a structure resembling a clover leaf? And this question was asked in the year 2004. Very easy question. The right answer is tRNA. Clover leaf, always remember tRNA is meant to be in that shape. tRNA, the other name is transfer RNA, occurs inside the ribosomes of the RNA, carries the information from DNA to the polypeptide, which includes hnRNA, that is the heterogeneous nuclear RNA, and the shape belongs or goes to tRNA. Next question here. Transformation experiment was first performed on which bacteria? So here they are talking about transformation. I think we already discussed this in part 1. We spoke about pneumonia, right? Transformation process was seen in pneumonia. Yes, the right answer goes for option B, Diplococcus pneumoniae. Transformation was carried out with the help of Griffith, and he used bacterial strains of pneumonia to carry out this experiment. Next question here. Extranuclear inheritance occurs in dash. So here you have a question, and it is associated with a killer strain in Paramecium. Color blindness, if you see, it follows a sex-linked inheritance, while if you have phenylketonuria, this is associated with a born error of metabolism. So there is different aspects, but when we talk about extracellular inheritance, it occurs in the killer strain of Paramecium, which is option A. Next question here. Due to discovery of which of the following in 1980, the evolution was termed as RNA World? Okay, this you might not know. I will explain you the answer. Okay, we have, the answer to this question is option C. Ribozymes are catalytically active RNA molecule which was discovered in the 1980s. They are self-splicing introns, right? They are self-splicing introns, indicating their possible role of intermediates in the evolution of biological system from abiotic substances. Therefore, the right answer is option C. RNA have enzymatic properties. So they used riboenzymes. So that discovery of riboenzymes only made this very popular during the 1980s. Therefore, the right answer is option C. Next question. The transfer RNA molecule in 3D appears dash. This is an easy question, and I think all of you will know what the answer is. Yes, it will appear in L-shaped. Okay. Kim and et al. Suggested that L-shaped model of tRNA to the X-ray diffraction method that they carried out, and this was a 3D appearance, and it appeared to be L-shaped, which was option A. Next question. In DNA, when AGCT occurs, their association is as per which of the following pairs? This is an easy question. You just have to analyze and tell me. What would be the answer? In DNA, adenine always pairs with thymine and guanine always pairs with cytosine, and these are called as complementary base pairing. Therefore, it will be option C, which is the right answer, because adenine is always with thymine and guanine are always with cytosine. Therefore, it is ATGC, the pairing system. Therefore, the right answer is option C. With this, we complete the part 2 of molecular inheritance. Hope to meet you in the next part with another set of questions and explanations. Thank you.